la serie temporal.
Ah, mira, de verdad.
De los pixels.
So let's start. It's a great pleasure to introduce Adrian Jacobo. Some of you know him already. For the younger one, probably not. He did the PhD here with us, under my supervision and peer supervision, working on optical pattern formation maybe at that time. This was 2009, so it was many years ago already. One of my first PhD right away. So after that he changed topic. He went to Dresden to the Max Planck Institute for Complex Systems. He moved to biology already there. He spent there a couple of 3 years. And then afterward he moved to the United States to Rockefeller University where he is a postdoc. And he has been working on developmental biology, mainly on the hearing system. And I believe that's related to what he's going to tell us today.
Thank you. Thanks, Damian. So yes, as you heard, all of this is Damian's and Per's fault. If there are complaints, talk to them. I started working, and I still work on pattern formation. As you can see here, mechanosensory epithelia, of which our ear is one of them, are a fantastic system to study pattern formation because these epithelia form patterns at many different scales, and these patterns are coordinated across scales, and they're very regular and they're really fascinating. So what you're seeing here is the surface of a bullfrog sacculus. So this organ is like an ear for frog. They use it to detect vibrations on the ground. So they put their bodies against the ground and detect low frequency vibrations. And they do this thanks to these cells, which are called hair cells. So what you're seeing here is the cells seen from the top. Each cell has what is called a hair bundle. So these hair bundles are very specialized structures and which enable the organ to convert vibrations in the fluid that surrounds them into electrical impulses. And I'll explain you in a second how they do that. But what I want you to focus on is into two things. So one is that each of these hair bundles that is made independently by each of the cells is a very regular structure with a predetermined number of these finger-like structures which are called stereocilia. And these form, if you get rid of them, I'll show a picture in the next slide, they form a very specific hexagonal pattern, which is something I worked on in the past. And also the cells form a hexagonal arrangement on the epithelia. Okay? And not only that, they all have the same polarity. So there's this tissue has this type of coordination where cells coordinate their positions on the tissue. They coordinate the polarity over long length scales, and they also inside each cell they can create patterns with a specific polarity and a specific arrangement.
Excuse me, what do you mean by polarity?
Polarity means that this structure is polarized in one direction, so it's taller towards this end, this side, yeah. And this is going to be important for the rest of the talk. So, and if you have questions about the system, just ask. So in this case, you can see that they have this bulb here, which is called the kinociliary bulb. It's always on the same side, and this gives them directional sensitivity. So this is very similar to the uticle and the cycle that we have in our inner ears. So this is how you detect accelerations. If you're in a car and you close your eyes, you know if the car is accelerating forward or backward because these cells have two different polarities. So there's a population of cells looking in one direction, there's another population of cells looking in the opposite direction, and they get activated differentially depending on the direction you move.
Where are they located?
So in the animal, they're located in the inner ear of the frog. So I don't know a lot about frog anatomy, so I wouldn't be able to tell you exactly. So they're in the head. But
they also hear the sound? The sound is also...
They hear very low frequency sounds.
Oh,
yeah. Mm-hmm. They have a papilla that hears also sort of lower frequencies, so they don't have high frequency hearing as we do. So these cells, called hair cells, are the same cells that we have in our inner ears. So in particular, for example, the cochlea is covered with these hair cells. So the cochlea has a membrane inside of this snail-like turn, And this surface is covered by hair cells, so this is the same schematic of this. You see the whole cell, this is the hair bundle, and this is the cell body that wasn't visible in the previous picture. And if you focus on the hair bundle, you see that it's made of these protrusions that are called stereocilia, as I said before. And these protrusions are linked to each other by what are called tipplings. These are chains of proteins. They are very remarkable in the sense that there's only one or two linking each of these, sort of the tall bundle to the short one, and they link the side of one of the stereocilia with a channel, with a mechanosensitive channel that is on the tip of the shorter one. So what happens is that when the fluid that surrounds the stereocilia deflects them, they shear, so they move, and the tipplings are put into tension, and then the channels, the mechanotransduction channels open, and the ions that surround the cell enter the cell gets depolarized, and then it releases neurotransmitters, so it creates a current, and the current is related to the brain. Okay, so this is the way that you are able to hear what I'm saying now. So this exact same process is happening in your ears now. If you close your eyes, you know that you're level to the ground because also the hair cells in your semicircular canals and your utricles are also undergoing this process. So it's very useful and very necessary sort of for having a decent life. And as I said before, so this is the same type of picture of a hair cell where we remove the stereocilia, and you can see that they're arranged in a hexagonal pattern. And this is something I work on, so how we make this pattern that is very regular but at scales of hundreds of nanometers. How do you generate this with enough precision? It's also something I worked on in the past. And so as I said before, so we have patterns that are sort of at the subcellular level, both sort of the arrangement of the stereocilia on the surface of the cell and the length of the stereocilia is very precisely regulated at the subcellular level. If we move to the cellular level, there's this long-range polarity. So in this case, you see that this black dot marks where this bulb is in each of the cells. So you see that there's two fields. So all of these cells point in one direction, all of these cells point in the opposite direction. So these are in mammalian vestibule, so they're used to detect linear accelerations in the head. And this one is a picture of what I'm going to be talking about for the rest of the talk, which is the zebrafish lateral line. It's an organ that is used to detect water currents. So the fish use them to know which way they're swimming. They know if there's other fish passing by, if the predator is trying to eat them. They use these cells to detect movements of the water, and they know what's happening around them. And as you can see, this organ is sort of a much simpler version of this organ in the sense that you have only two rows of cells and they're opposite in polarity. So this black dot marks sort of the polarity, the orientation of the cell. So you see that there's this green thing and the black dot on each side. So it forms a mirror symmetric arrangement. So the cells are facing each other and you have this equivalent populations of cells of each polarity. Okay? And then, so if you go to the tissue level, as I said before, you have sort of these, here in this picture, the red ones are the hair cells and the green ones are support cells that surround them. And they form this lattice in which there's never a hair cell touching another hair cell, so there's always a support cell in between. And depending on the tissue, for example, here, this is a picture of the cochlea, so they form this sort of square-like lattice. And so the red ones are the hair cells, and they're always separated by support cells, and they form this very regular pattern of 3+1 rows. So as you can see, there's a lot of places where one can study pattern formation, and there's a lot of different processes that give rise to patterns depending on the scale. So we think that at this scale, there's a Turing-like pattern that is sort of a true Turing-like because it's a reaction diffusion equation happening here inside the cell, and in this case it's something like a Turing pattern, but it happens in between cells, so there's not a fluid in between, so there's some interchange, so the cells act as units of the pattern, and so on. So what I want to focus on for the rest of the talk is on this organ, the lateral line. So this is a schematic of a 2-day-old zebrafish larvae. These are like tiny tadpoles, like frog tadpoles that you can see in a pool or a pool of water. So these organs are marked in green, so they run along the whole fish, and they're right underneath the skin. So there's like in a human would be so right underneath the skin and protruding outside the skin of the fish. And they form like several lines of these organs, that's what is called lateral line. And each one of these green dots is formed by, it's what is called a neuromast. So the neuromast is the organ itself, each of these units of the lateral line. So you can see here a side view. So these are the hair cells. So they have like these tiny bundles, but they're similar to this picture here. So each one of these is a hair cell. They're separated from each other by these support cells, which are marked in black here, because I mark them in black because in the usual pictures that we use here, we don't see them. And you can see here the bundles, which are these tiny green structures here, and seen from the top they look like this. Again, they have a polarity which is marked by this black dot. Okay, so these organs have these very long cilia that stick out of the skin of the fish, and they're in contact with water. So when water deflects the cilia, then the mechanical force is transmitted to the bundle, and then the bundle opens or closes the channels, and then this depolarizes the cell, and then the fish knows if there are water currents moving forward or backward, and then they have another population that is exactly the same, rotated 90 degrees, so they can detect also dorsal-ventral, so up and down flows of water. And in particular, they have a very strong response if you deflect the bundles that are facing towards the tail, because this is the first thing they want to do, because predators come from behind and suck water and suck the fish in. So if you try to grab them with a pipette or something and suck them from behind, they have very strong responses. The first thing they do is they swim away, like as fast as they can. What we want to understand, so this is sort of a very simplified version of these complicated organs. So it's a nice system to study because of many things. So one is that you can look at these cells in a live organism intact. You can put them under a microscope, and because they're right underneath the skin, you can see them. The zebrafish are also very amenable to do genetic manipulation. So in this case, for example, these cells have a green protein that is a transgenic protein that we can insert in the fish and label the cells. And so they're fast to breed, so they're very convenient as an experimental organism. And also the organ is sort of the simplest set of organization of hair cells that you can find. So in that sense, it's sort of like a hydrogen atom of morphogenesis of this epithelia. So what we want to understand is how this organ achieves this mirror-symmetric organization of hair cells. So first of all, how does it know how to make the same amount of hair cells of each polarity, and how does it know where to put them so they're facing each other? Okay. Any questions so far? So I'm going to move to try to explain sort of the first question, which is why hair cells come in pairs. Okay. So they come in pairs. The question is kind of simple, is because they're created in pairs. Okay, so so what you're seeing here is it's cut through this plane in the organ. So these are real cells which we mark with a transgenic protein that is green under the microscope. So you see all the cells that are already mature, and you will see here there's a cell that is unlabeled that will decide to become a pair of hair cells. So when it decides to become a pair of hair cells, it upregulates these green fluorescent proteins, so it will become visible. Okay, so I'm going to play the movie, and you will see here that there's this blob that's starting to become sort of shade of green. Okay, so there's a cell that
this is seen from the top.
From the top, yeah. This is seen from the top.
Large, seen from the top.
Yeah. So it became this sort of. It was a stem cell-like population. It becomes a transit amplifying cell. So it decides it wants to become a hair cell. And then it divides in two, so you're going to see it divide now here. So now there's two cells there, okay? And And when it divides, sometimes, and I'll talk about this later in the talk, in the second part of the talk, they undergo these coordinated rotations. So you see that the cells exchange their positions. I'm going to play it again. So you see that the pink one sort of moves to the place of the cyan one and vice versa. And then after they do that, they sort of go and mature, become mature hair cells, and in particular what they do is at this point they're sort of round and they don't have a hair bundle at this point. So if we move up in the focal plane up where all the hair cells have their bundles, okay, you see that there's nothing here, and what you're going to see now when I play the movie is that you will see that they will develop these surfaces, and the surfaces initially are round and depolarized, and then they become polarized. So you see that they have an orientation, okay? So you see that this black dot sort of appears in one end. So you see now these two round surfaces, and they're sort of not polarized, and now they're starting to become more and more polarized, and you see here sort of this black dot, which is called the kinocilium. So it's on one end, and they're facing each other, okay? So you see it when the movie stops, it will be more clear. You can see it, so you see there, this cell has a black dot here, and this other cell has a black dot on the other end, okay? So kind of like this schematic. So the question on how the cells are made in equal numbers is easy, so you make one of each, okay? So that's easy, so now the thing is, how do you know that you're making one of each? How each cell knows the polarity of the sister in such a way that they can control for this, okay? So a partial answer for this was given a few years ago when people found that there's a gene that is expressed that is called EMX2 that is only expressed in half of the cells, okay? So when a cell expresses this gene, the bundles are facing one direction. When it doesn't express it, they're facing in the opposite direction, okay? And the way that it works, I'm not going to go into the details for this, but the way that it works is that these cells have sort of a kind of nematic field that is given by this protein bundle 2 and some others. So this field is always polarized in the same side of the cell, and then depending on whether this switch is on or off, the hair bundle will develop facing the field or facing opposite from the field, okay? So the cells know their orientation, and then they have some internal sign, so some internal polarity. So they say, if I'm positive, I'm oriented in the same direction as the external field. If I'm negative, I'm oriented in the opposite direction. So they're kind of, you can think about them as dipoles with a sign. Okay?
So is this nematic liquid crystal or
so
nematic or have polarity?
It has a polarity for the external field, but for the cells, it doesn't have a polarity. So the cells have their own polarity that says if it's positive or it's negative. So the analogy is not complete, and it depends on how you write the theory, whether you interpret this as a nematic field or not. Yeah. So, but yeah, so I wouldn't say it's fully nematic because it does have a polarity. So the thing is, what is the cell seeing? That is the cell seeing the polarity or not? And so in some sense, you can say that they're not seeing the polarity, and the polarity is just an intrinsic property of the cell. There's a C in a direction, okay? And then they decide whether to face each other or not.
Sorry, what is this external field? Gravity or?
No, there's no one really knows. So there's many things. So there's many things. So one is that there's gradients of proteins across the tissue. And these gradients give a polarity, but there's many redundancies, so there's not just one thing. But what they do is that these gradients then orient the localization of these proteins on one side. So this creates another field that gives a polarity. But this is also redundant. So the cells, the way in which this organization is created is like a domino. So first the cell will polarize its proteins, and then this will make these proteins polarize on the other end, and so on. So if you knock out the external field, what they do is, because you have a small organ and you have noise, so essentially the orientation will curve and you will have a spiral, essentially, of the cells are sort of trying to orient with the nearest neighbors, and they're kind of making tiny errors, and they're following each other sort of in a spiral. And if you have a bigger field, so this is a very well-conserved system that orients cells in the skin, orients cells in wings of the fly and many other organs. So depending on the size of the system, you see different effects. So if you have a very big system, you just see sort of random meandering of the polarity locally if you ablate the long range order. If you have a small system like this, you're dominated by boundary effects, so you see just spirals. Yeah. But yeah, it's very redundant, and there's many gradients across the organ that sort of give this orientation. So if you knock out one or two genes, you still have orientation, and yeah, so it's a complicated system. I mean, it's very vital. So if you knock out these proteins, so if you have mutations on these proteins, usually you don't survive very long. So most, like for example, the fish only survive a couple of days, the ones that have mutations on these genes, because many of the organs depend on the right orientation of many cells. So it's what I want to say with that is it's a very well conserved system because you don't want it to go wrong. And this is just to say that you have equal numbers of cells of each polarity, and you can see that the cells that are in one polarity, which I'm going to call caudad from now on, so cells that are pointing towards the tail of the fish, are positive in the EMX2 gene, and the ones that are rostral are negative. And this is not only correlation, so it's not that the cells are correlated with the expression of EMX2. If you flood all of the cells, if you create a transgenic fish where all the cells have this EMX2 factor, all the cells are pointing in the caudal direction towards the tail. And if you mutate the gene so that the protein is not functional, they point in the opposite direction. So you can see that this gene does control the polarity. But this gene is intrinsic to the cells. So this gene doesn't transmit any information to other cells in any way so that they can know what the polarity is of the system. Okay? So there has to be something else where the cells are transmitting some information in some way that they can use to coordinate what is happening. Okay? So to explain you what that is, I need to give you a short introduction to what is called the Notch signaling pathway. So this pathway is it's simply a cascade of molecules that send information from the outside of the cell to the inside of the cell. In this case, this Notch pathway is used for cells to communicate with each other at short distances. So this is a pathway where you express some proteins on the membrane. These proteins that you express are called Notch and delta, and the proteins interact with the proteins of your neighbors. Okay? So if you have a protein of Notch that interacts with the protein delta that your neighbor has on the surface, your Notch protein releases a piece which is called the Notch intracellular domain, NICD for short. This piece goes to the nucleus and then it can bind to DNA and affect the expression of other genes. Okay? And in particular, what it does is that it creates positive feedback for Notch. So if you have Notch, you create more Notch. And it downregulates, it inhibits the production of delta. So if you have more Notch, you create less delta on your cell. Okay? And these I'll go into details in a couple of slides. But what is important is that this creates a positive feedback between the two cells. So this creates a nonlinearity where there's a winner-take-all kind of mechanism. So if this cell has a lot of Notch, it will make this cell produce less Notch, and it will suppress it for the production of Notch. Okay, so it creates a symmetry breaker mechanism that I'll explain in a lot more detail in a second. But so the only thing that you need to know here is that when Notch gets what is called cleaved, so it releases this piece to the cytoplasm, and then can bind to DNA. It can act on other genes. Okay. So the first indication that we had that this type of cell-cell communication was involved in this process were some experiments done by a former postdoc in the lab where he used this molecule that is called DAPT. This molecule, what it does is that it inhibits the release of the Notch intracellular domain when there's an interaction. Okay? So when this happens, the cells essentially are now isolated from each other. They can't talk anymore because this is the signal that you need to send to produce any effect. So if you're blocking this, the cells sort of meet each other, but they can't talk. They're sort of separated from each other. So if you do that, what you see is that you get an excess of cells of one polarity, in this case of rostral polarity. Okay? So instead of having 50-50 like we had before, so now we have more cells of one polarity than the other. Okay? So this gave us the idea that, okay, well, it seems that this notch pathway is involved in some way. So the easiest way to think about it was, okay, if we are blocking notch, then the cells will have no notch, and then they're obtaining one polarity. Okay? So the logical assumption is, okay, what happens now if we put a lot of notch inside the cells? Presumably they should do the opposite. So you should have a lot of cells of the opposite polarity. Okay? So we went and did this experiment. I did this experiment for those of you who know who knew me during my PhD. So we went and looked, and what happens is that you have either, in this case, so these cells that are cyan are expressing this notch intercellular domain all the time. So this is sort of turned on like an external forcing, is turned on all the time. And when this happens, you get even more cells that are of the same polarity that when you block notch. Okay? So at this point we were very confused. We were like, okay, what's happening? So if we have no notch, you get one thing. If you have a lot of notch, you get the same thing. So even stronger. So what is it happening? And so here is where being a student of Per and Anya pans out, because you know that if you have a nonlinear system, you should never trust your gut. So you should always look at the bifurcation diagram. So I went and looked for a model that described this system. So this is sort of a single model of 6 differential equations where you have a term that describes the production of notch, so this molecule here for cell 1 and cell 2. A term that describes the production of delta for cell 1 and cell 2, and a term that describes the production of the notch intercellular domain for both cells. Again. And this tells you, this term here tells you that if you have a lot of, so the notch intercellular domain creates more of notch, it suppresses the production of delta, so these are essentially just sigmoidal functions with opposite signs. So this creates more, this creates less if you have the notch intercellular domain. And then you have these two interaction terms that say if notch of one protein interacts with delta of the other protein, they create one molecule of the notch intercellular domain. Okay? And then you have decay and sort of, yeah, so it's nothing too fancy. Okay? So if you look at these equations, what you can see essentially is, as I said before, this describes a positive feedback between the two cells. Okay, so if a cell has delta, it will make the other cell produce more notch, and in turn it will suppress its own delta, which will create more notch. Okay, so essentially if you go from notch to notch, there's a repression, and if you go from notch of one cell, there's repression of notch of the other cell, and vice versa. Okay, so each cell, it's a competition, they're trying to inhibit each other. So here, if you start from a situation where you have a stochastic, so you have essentially zero amount of protein and just stochastic fluctuations between the two cells, one will win the competition, will go to a high notch level, while the other one goes to a low notch level, and the same, so the opposite is true for delta, and so something similar happens for the notch intracellular domain. Okay, so if we had a system like this, you have competition, you have bistability, so one of the cells goes to a high level, the other one goes to a low level, and then you can use these to make decisions, okay? So now, if you assume there's a threshold, if the cell that is above the threshold will acquire one polarity, the one that is below the threshold will acquire the opposite polarity, okay? And at this point, the model is sort of ambiguous with respect to which of these states corresponds to which of the polarities, okay? So to disambiguate the model, we need to go back to and look at experiments. So we can make a bifurcation diagram and look at what happens now if we add this extra term that is a constant expression of the notch intracellular domain. Okay? So now we turn this on, which is the same thing we're doing for our transgenic fish. You turn it on, and then you see that for sort of low values of this term, you still get these bistable steady state, but then at some point you get to a sudden node bifurcation where the steady state disappears, and then you get just a symmetric state where both cells have very high notch. Okay, And in particular, so if you have the threshold set somewhere around here, you see that immediately both cells are above the threshold, so they should both have the same polarity, which is what we expect. And if we look at the experiments, we know that the cells that have a lot of notch, they have rostral polarity. Okay? So this disaggregates the system, and it says if you're above the threshold, you have high notch, you get rostral polarity. If you're below the threshold, you get rostral polarity. Okay? So now we can try to see what happens with this inhibitor that we were talking about before. So why now if we inhibit the amount of notch, we also see cells that are in this high notch state. Okay? So what happens is, again, so here what we can do is create a bifurcation diagram changing this constant Kt. This constant is how much notch is cleaved, so how much of notch is released into the cell when notch meets with a delta molecule. Okay, so the lower the rate, the lower. So the more that you inhibit with this drug, the lower this rate is. Okay, so going in this direction in the diagram means adding more and more of this compound that inhibits notch. So what you see here is that, of course, if you have zeros, or if you're in the sort of wild type level, you have these bistabilities, so you have high state and low state. But then there's a pitchfork bifurcation here, where a subcritical pitchfork where you create this symmetric state, okay? And this symmetric state initially is above the threshold. Okay? So what happens is that you start inhibiting, and then some of the cells, the ones that have initial conditions are in the what's the name? So attraction basing of this branch here, they fall into the symmetric branch. So we both have the same amount of notch, but this amount of notch is enough to make them polarize in the rostral direction. Okay? So what happens? So what this tells you is that if you were able to increase, so to decrease this value of Kt even more, which would mean increase the concentration of the drug, you have to reach a point where the symmetric state is below the threshold, so you should see all the cells becoming polarized in the opposite direction, the caudal direction. Okay? So at this point where we had these results, we couldn't use this drug we were using, the APT, because it's toxic for the fish. So they don't survive, so you get no cells at all. So what we did is we found a mutant that lacks this notch protein. So it has absolutely no notch protein, so that means that Kt is 0, there's nothing to cleave, there's nothing. So these fish sit on this corner of the bifurcation diagram, so there's no excuse here. There's nothing that can happen to make these cells point in the opposite direction, so they all should be caudad. We're very nervous when we got the fish, but when we look at them, they're absolutely all of them pointing in the caudal direction. Okay? So this sort of seems to imply that if you, essentially if you're at this point, if you have absolutely no notch, then you get caudal direction, and when you get a lot of notch, you get the opposite direction. So this, so it seems that it really is sort of this bistable system where the cells sort of compete with each other, and you get either one polarity or the other. And after we did this, we found another compound that is that has a stronger affinity to block notch, and so you can use at lower concentrations, you can see the same effects. So if you use now no compound, so this is just the control, what you see is the cells are 50-50 polarized, as in the wild type, as before. When you add so the intermediate concentration of the compound, you see a bias in the rostral direction. So you see more cells, in this case marked with blue than red. And if you keep increasing, you lose the bias again, because now this middle branch is sort of at the same level of the threshold, so the cells randomly will go both in one direction or the other. But now if we increase even more the amount of the compound, you see now that you can reverse the bias and make them point in the opposite direction. Okay? So now by changing this other drug, and just as a reference, the original one, the APT that we were using, is sort of here in this blue region of the diagram. So by using this drug that has a high affinity, we can sort of transverse the whole bifurcation diagram and see its effects. Okay? So
why didn't they not sum 100 is not tough enough?
Because some of the cells are too young and we can't tell which direction they're facing, so we don't score them. And it's a way to see this notch pathway also controls how the cells decide to become hair cells. So if you block it, you see many, many more hair cells. So this is sort of a way to quantify that. If you have a lot of hair cells and you can't tell the polarities, it means there's something sort of wrong happening in the organ. So the next thing is that, as we said before, there's this molecule that is called EMX2 that is expressed only on the cells that are caudad in polarity. So what we found is that Notch controls the expression of EMX2. So Notch inhibits the expression of NICD. So in these cases where you have these blue cells that have this extra Notch term, then they have no EMX2. And in this case where you have the mutant, all the cells have EMX2. Okay? So this Notch seems to just be a master regulator of what EMX2 is doing. And this sort of tells the cells what they have to do with their polarities. Or so we thought. So the idea here is, so this would be sort of a simplified diagram. So Notch controls EMX2, and then EMX2 is sort of a passive readout of what the polarity is going to be. So if it's on, you do one thing, if it's off, you do the opposite. But this means that if we now fix the concentration of EMX2 to a given value, the cells shouldn't care what NICD is doing, okay? Because this is the final readout, okay? So you can sort of model this and the concentration of EMX2, so EMX2 is just a passive readout of what NICD is doing, okay? And nothing else should happen. So we want to leave this experiment. So now we overexpress EMX2 and overexpress NICD. What you should see is that all the cells are caudal like the EMX2 overexpression, but what you see is that they're instead all rostral, okay? So it seems that this is not just a passive regulation, but that there is some competition where sort of if you overexpress both, NICD seems to win. So this is very similar to just expressing one of them and is overexpressing both of them. So it seems that there's some competition where this tends to win over the aggregation of EMX2. And I'm not going to go into detail, so I can move on to the second part of the talk. I'm not going to go into much details, but we can model this as a system where you have interaction, so downregulation of EMX2 by NICD, but then they compete for some unknown gene or set of genes, which we call the polarity effector, that is the one that in the end decides the polarity. Okay? And then so you can simulate this type of system, and you can see sort of you can recapitulate the wild type experiment. So this is the concentration of EMX2, this is the concentration of NICD. So you have a state that is high in NICD and low in EMX2, and then you have one polarity, the blue one. And then the opposite, if you have low NICD, you have high EMX2, and then you have the opposite polarity, caudal. And what happens, so is that this purple line here sets you can calculate what the threshold would be. So, and this line sets the threshold, and you see sort of the cells are above the threshold get one polarity, the ones are below the threshold get the opposite polarity, and we can simulate now add these constant terms for both NICD and EMX2, and we can simulate what happens there and sort of recapitulate what we see experimentally. Okay? And this is sort of now work in progress that we're trying to identify what this gene might be, or genes, and how the sort of the actual regulatory network might look like. So this is sort of the simplest model, and I'm sure it's not going to be like this in the end. So now I want to move to the second part of the talk, which is that if you were paying attention, you probably know this, that as I said that this is a stochastic process. So you start from two cells that are symmetric. Because of stochastic fluctuations, they break the symmetry, and then you end up with one cell of one polarity and the other cell of the opposite polarity. Okay? But this means that you can end in either this situation where both cells are facing each other, okay, or in the opposite situation in which the cells are facing away from each other. Okay? But in the real system, you never see this configuration here. Okay? So the question is why? So in the real system, they're always mirror symmetric and always facing each other. So you never see these. And so the question is why they do this. Okay? So one thing that I need to tell you to understand how this happens is, first of all, that these cells have some protrusions that are oriented in the same direction as their polarity. So when they're developing, you will see this movie now. I'm going to play it, and you'll see these two cells. So it's not super clear. Can we maybe turn off the light? Yeah, maybe I'll play it again. So you'll see that the cells become sort of extended in one direction. So you see here, they become extended in one direction. And this direction sort of coincides with the polarity that the bundle is going to have. Okay? And the other thing is that they sort of move in the direction of these protrusions. So they're kind of crawling, using these protrusions to crawl, and they crawl away from each other in this case. Okay? So if you look at the direction of the protrusions, you see this protrusion here and this one there. The direction of the protrusion is correlated to the direction of the bundle. So the cells have some internal compass telling them which way is left, which way is right, and they're able to reorganize their whole bodies in that direction, and then organize the bundles after. When they're mature, they can organize the bundles also in the same direction. Okay? And these two things are correlated. And if we look now at these notch mutants where all the cells are oriented in the same direction, we also see that the protrusions, like you'll see in these two cells here, so they're both pointing in the same direction. So this one's pointing this way, this one is pointing that way. Okay? And you can quantify this, and you see sort of that the protrusions, as the bundles point always in the same direction, the protrusions also point in the same direction. Okay? And these protrusions, as I said, they're correlated with cell movements. Okay? So the cells move in the direction of the protrusions. So what happens is that if you have cells that have opposite directions, the center of mass of the cell pair remains constant, but the cells move away from each other. So this is the distance between cells, and they move away from each other. Okay? And at the same time, the center of mass remains constant. And if you have cells of the mutant fish where they're all moving in the same direction, the cell distance remains constant, so they don't separate, but they do translate together. Okay? So essentially what's happening is that if you have cells of opposite polarities, they move away. If you have cells of the same polarity, they move together. Okay? So what happens is that now you're linking the polarity of the cell and this internal state of whether they're Notch positive or EMX2 positive to the movement of the cells. And if you do that, what you find is that if you get trapped in one of these configurations in which the cells would end up facing away from each other, what they would do is they would try to move past each other. So you see these two, so if they differentiate, they divide there, and now they grab each other and they tumble. Okay? So each of the cells is trying to move. I'll play it again. So essentially what happens is that this cell, because its polarity is facing this way, it will try to move in that direction. This cell has its polarity facing the other way, it will try to move in the opposite direction. And because they do this and they're attached to each other, they sort of tumble. So it's like if you're in a revolving door, you're trying to get in, someone's trying to get out, and you just make a torque on the door and you just flip. Okay? And then you end up on the outside, the other person ends up on the inside. Okay? And the cells do exactly the same thing. Okay? And so what we see from these is that not only these movements correlate polarity with cell movement, but they also happen 50% of the time. Okay? So they happen 50% of the time because this is what you would expect from this unbiased stochastic process. So 50% of the time the cells will get it wrong, will get the wrong configuration, and when they do this, they rearrange so that they can fix it. Okay? Yeah, as you can see, so you can interpret this as a dipole on an external field, but in this case, it's not the field that is generating the force on the dipole, it's the cells, so it's the dipole itself that generates the force, so the cells are the ones that are moving. So they're sensing this external field and they're moving. And what you see is, again, so if you start from this configuration that is wrong, so the blue cell now is on the left and the red cell is on the right, they sort of move around, they generate these linear forces, they move around, and they end up in the opposite configuration where the red cell is on the left and the blue cell is on the right. Okay? And one can understand sort of the kinematics of this movement first by assuming that they generate a constant force. So if we assume that they're like a linear motor generating the constant force against the drag, if you do this, you can calculate the rotation, the angular velocity of the pair depends on the angle as a sine of the rotated angle, and we can sort of match these to to our measurements of the rotations, and we can do something a little bit more sophisticated, which is we can write the surface energy of the cells. So we can imagine that these cells are kind of like soap bubbles, so they have a surface tension. So they have a surface tension that creates an adhesion energy between two cells, and then they have different adhesion energies with the surrounding tissue. Okay, so all the other cells which are sort of swimming around that you don't see, that are always in black in these movies, they create this sort of sea in which they're swimming, and they have an effective energy which might be different for each of the two cells. So in this case, so if you write this interaction energy, and then you assume that these cells have an internal gradient of surface tension, you can calculate exactly the same thing. So this gives you these linear gradients in this energy give you a cosine shape potential. So essentially, what it tells you is that the state in which the cells are, so the positive one is on the left and the negative one is on the right, is an unstable configuration. So they will always fall into this configuration, which is the stable one. That is, so the cells facing each other. Okay, and we can calculate again. So if you calculate this energy, you can calculate the same thing. The rotation speed is again depending on the sine of the angle, which is the same result we got by assuming that the force is constant. Okay, so we can describe this using this effective energy theory, which also has so it makes the prediction that because you're coupling polarity to cell movement, the cells of the mutants that always have the same polarity should never rearrange. You should never see one cell jumping on the other, so they should always move in the same direction, and this is what we see. So in the control, so the wild type, they rearrange 50% of the time, and the mutants, they never do. Okay. So just to summarize, so what happens in this system, the way that we create this mirror symmetric organ is by initially you have a biochemical state that breaks the initial symmetry. So you have sort of a double weld potential. You can fall into either of the two configurations, and so one of these configurations corresponds to the one you want to be in, which is the cells facing each other. The other configuration is a configuration you want to get rid of. And then what you do is by linking the biochemistry, so linking this internal state of the cells to the cell movement, to the mechanics, you break the symmetry, so you tilt the landscape, and you create only one minimum where the cells are going to fall, and you always end up, regardless of how you start, you always will end up with this final configuration of the two cells polarized in opposite polarities and facing each other. So with that, I would like to thank Jim, my supervisor, Agnik and Anna, and my collaborators, and Kim, who started this project a long time ago, and my funding sources, and you.
Any question? I have one myself. It's from the first part when you had when you showed this you show a place where there were in a tissue there were half on one side, half pointing on one side.
Yeah. You draw a line. Yes.
Exactly. So what was that? Is that stationary thing or something that can flip and relate to one?
No, that's stationary, yeah. So we don't know exactly how this is created, because in our case, so this is something I didn't mention, but one of the reasons why we study this organ is because, so if there's one thing that you're gonna take home from this cell, it's from this talk, is that our hair cells never regenerate. So if you lose them, and you lose them with age, you go deaf and there's no going back. So if you're going to a concert, wear earplugs. But the fish are able to, these cells are in contact with water, and they're always in a very abrasive environment, so they die and are replaced all the time. So the fish know how to do this. They know how to replace them, and this is one of the reasons why we study this system, is because we want to understand how they do this and how they maintain sort of the same tissue architecture, although you're replacing the cells all the time. So these cells are from a mammal and they're not replaced. So this is created as a crystal essentially. So you create it during development and it's fixed. And we don't know how it's made because in this case you can do it by local coordination, but in this case you have to do long-range coordination of cells. Yeah. So initially we had the idea that this might be created sort of like as like a tectonic plate. So you're creating cells at the center and then they're displaced. But this doesn't seem to be the case. So there seems to be something that creates this line and then propagates or something. We don't know. Yeah. So there's nothing. No, there's not much known how it's done, and I think it's super interesting. Yeah.
It's probably some special effects. There's some the phase or long range.
Yeah. So I'm sure that this notch pathway is involved in some way, but I don't know how you translate these local interactions into long range. Yeah.
Very interesting. This, and we will ask about this mutually repressive notch interaction. Because in one of the papers that you mentioned, the sprint sac in Nature 2005, in the lab of right? They observed this kind of black and paper pattern.
Yes.
It's called biology, but you seem to have only this small arrangement. I mean, you have a bigger.
Yeah. So this is because the cells we don't fully understand it, but what we think is that these cells when they're mature, they all have apical surfaces and they're all touching on the apical surfaces. But when they divide, what they do is that a cell one of these blood cells will retract this apical protrusion, will become round and more basal. It will divide, they will undergo this signaling, and then they will shoot apical protrusions again. So what we think is happening is that all the signaling is for these cells is happening up here, and then these two cells are isolated from everything else, and they just signal to each other. So in that sense, it's a neat system also to study notch signaling because you don't have these long-range coordination effects. So you don't have this salt and pepper so there's a lot of problems with this model for salt and pepper because if you have an infinite time, you converge to something that looks sort of hexagonal or salt and pepper, but if you have a limited time, and that's always the case with organisms, it's very hard to converge, especially because the convergence time scales like n squared. So as soon as you have many cells, this is not very feasible that's the only thing that's happening. So in this case, this is sort of like a hydrogen atom of the notch interaction because it only happens between two cells and two cells only. And so we can see that we have experiments where we can kill one of the cells during the process, and then we know what's going to happen to the other cell because you lose your signaling partner. So we know that they're only doing this with each other, essentially. So you lose the signaling partner, and because you have no signaling, then the other cell always adopts the same polarity. So we know that this is localized. So what we want to know is to show that this is where notch is expressed is sort of in this compartment, and this is happening sort of separate, spatially separate from the rest of the signal.
For the audience, maybe say because people have listened salt and pepper. What is this salt and pepper? Biologists like to say when you have.
Yeah, so I can show. Yeah, so these, for example, is what Manuel is referring to. So in these dark areas here, there's other cells that are called support cells, and this form this salt and pepper pattern in the sense that you have one cell that is surrounded by cells of other types. So this red cell is surrounded by blood cells, and this red cell is surrounded by blood cells, and they never touch. So the red cells never touch each other. And this is, in this type of tissue, is very important. So if two red cells touch each other, they die. They don't survive. So if you have, for example, a lack of blood cells, which are called support cells, then your cells die and you go dead. So this is a very important process for maintaining the tissue architecture.
Okay, so thank you very much. I think we're going to thank everyone again.
Visto que todas las mayoría de las sesiones.